I go by Tapa. I'm a postdoc here at Princeton. I work with Professor Sujit Datta. I am joined by two other grad students from the Sujit Datta lab, Jenna and Daniel. Jenna and Daniel, please introduce yourself.
Hi, I'm Daniel. I'm a third-year grad student in the lab.
Hi, I'm Jenna, and I'm a second-year grad student in the lab.
So what we plan to do is we'll take, each person will take turn, answer a few questions, and I think you can also send your questions to the Q&A. So we are ready, we should go ahead and start the session. Josh?
Go for it.
All right, sweet. So I'll go first, and then we'll take turns to answer your questions. Okay, cool. Let's see. Okay, here's going to where I want it to go. Okay, cool. So the first question that comes to us is why E. coli? Why we care about E. coli? Why it is important to study E. coli motility? The simple answer is it's a very well-studied bacterial system with well-known signaling pathways. People have studied that for years. And why? Because they normally live in the intestines of people and animal and responsible for many infections. So people started studying them, and you can read this paper where John discusses E. coli as an experimental organism, why it's important. It's actually a preferred host for many gene cloning due to high efficiency of introducing DNA into the cell. It's also used to produce proteins because of its rapid growth and ability to express different proteins. People used to transfer large DNA fragments into from one bacterium to another. It's also used to study how metabolic pathways of this bacteria work. So when we are studying motility, since we have this large pool of information, it's easier for any researchers to understand how this signaling pathways are controlling the motility. That's why E. coli is very well studied in this form. The next couple questions that we got were how a flagellar motor torque related to the speed of motion, or what kind of understanding do we have about the flagellar motor? So the bacteria actually rotates the flagella by pumping out ions, right? The motor is powered by the flow of ions through an electrochemical gradient across the cytoplasmic membrane. So it depends on different proton motive force, which is basically a sum of the membrane potential, between the inner side of the membrane and the outer side, and the transmembrane ion concentration gradient, which you control by changing the pH. So if you fix a proton motive force by forcing the pH, you can attach a small tracer particles to one of the flagella, and you can measure how fast this flagella is moving by measuring how fast this bead is rotating. And how do you change the drag coefficient, which is given by this side? Drag is given by 6 pi eta r. R is the radius of the bead, and eta is the viscosity of the fluid media. So as this bead rotates and you measure how fast it's rotating by measuring the angular speed of the rotation, you can calculate the torque of that flagella. And if you plot the torque at different angular speed, you'll see that until a constant, until a speed which is given by this ink, it's kind of constant, and then it starts to decay with the increase in speed. So there is this paper actually discusses the concept in detail. So if you're more interested in that, you can go and read this paper.
Hey, Tapa, you got a question in the Q&A. The question is, how is the membrane potential different than the ion concentration gradient?
Okay, so the membrane potential, in my view, is the charge of the inner membrane and the outer membrane, and the ion concentration gradient is the ion inside the cell and outside the cell. Jenna, do you have any more questions?
No, that was it.
Okay, the next set of questions where can you explain how signaling pathways happen downstream of the TAR receptor, which I believe is once the aspartate binds to TAR, how the signaling actually controls the flagellar rotation or chemosignaling. And then there is another question of how adaptation is necessary, or like how the adaptation work in this sensory network. So when aspartate comes and binds to this TAR, the TAR actually reacts with, it actually reacts the Ka, this A molecule. And this A molecule is actually responsible to interact with Y molecule and phosphorylizing this Y molecule to YP. So when this YP becomes phosphorylated, it goes to the motor and it changes the rotation of from counterclockwise to a clockwise direction, right? So cells, they start running and they become they start to tumble. Now, when aspartate binds to TAR, it reduces the catalytic activity of A, which acts as a catalyst in this Y to YP reaction. And it reduces the concentration of YP. And YP is also being constantly degraded to Y by Z. So what happens is that there is an overall decrease in YP concentration inside the cell. So cells are more probable to remain in the flagellar rotation, are more probable to remain in the counterclockwise rotation, and thus it keeps running towards the more aspartate rich region. That's how the chemosignaling works. Now, what is adaptation here? So remember, there are two A molecules here, right? So once aspartate attaches to the TAR, this A is also responsible for this catalytic activity of this reaction. Now, there are two things happening here. B becomes phosphorylated in this reaction, and it immediately degrades to B again. And meanwhile, it converts this methyl to methyl alcohol. Because like a reduction of catalytic activity of A reduces the concentration of this phosphorylated BP, this R molecule, which actually inverts this methyl alcohol to methylene group, starts to methylate this TAR, which reduces the catalytic activity of A again. That's how cells, if you put the cells in a higher aspartate concentration, eventually they will get used to this higher aspartate concentration, and this they will adjust the catalytic activity of A. But if you put in a gradient of aspartate, then the cells start to sense the more and more concentration of aspartate and start running towards those higher aspartate concentration by elongating their runs. all right, this is, okay, cool. So then there were a few questions about E. coli motility in porous media. So there is a general question, why quadratically varying MSD is indication of a ballistic motion. So if you measure the root mean square speed, the root mean square speed is given by the square root of mean square displacement over the lag time, which is the time difference that we use to measure the speed. So when MSD scales as or has a time dependence which is squared, quadratically varies with the lag time, the V is actually a constant speed, right? It's time independent, so which refers to ballistic motion. What is the definition of super and subdiffusive motion? The diffusive motion is where mean square displacement is a linear function of the time, which means your displacement is actually square root of time, which is by definition is diffusive motion. Now subdiffusive means your effective displacement is less than the diffusive motion, where this exponent of tau is between 0 and 1. Super diffusive motion is where this exponent is between 1 and 2. And remember, when this exponent is 2, then it becomes ballistic. Okay. There was a question between how do we compare hopping and running. In both cases, actually, flagellar remain bundled. But runs are controlled by the flagellar rotation only, where hops are controlled by flagellar rotation and availability of directed pathway. Sujit will show you an example in his talk that where flagella remains bundled, but the cell is inside a trap, which means when the cell should have been running in liquid media, in porous media, they are trapped, they can't run because they don't have a space to go. Porous media with spatial gradients of hydrogel of different densities to differentiate cellular motility. It's a great idea. The obvious challenge is if you use the same hydrogel particles and pack them really close to each other, they can swell and deswell to an average size by osmotic pressure balance. And why we want to do this? Because we want to know the transient motion at the boundary of two different pore sizes, which is a great idea to explore, but for our purposes, we are interested to see at the steady state motion at different pore sizes. So we can get the same information by separately studying different porous media. Is there a way to replicate hopping trapping on other type of bacteria? Absolutely. We have tested two other different cells, Vibrio and Pseudomonas. This work is currently under process, so not much information to show right now. I think there was one question about why longest run in bulk unconfined liquid is an order of magnitude shorter. I believe when we are talking about this, we are talking about the run duration, which is order of 1 to 2 seconds. When compared to a trap duration, maximum duration we recorded were about 40 seconds, which is an order of magnitude larger than the run duration. Even like the hopping durations are way smaller than the run duration. The point we want to make here is that trap durations are the most important while determining bacterial diffusivity. But in liquid media, diffusivity is given by the run speed and the run length. So the run duration is more important in liquid media. Because the tumbles are instantaneous, but here traps are not. Traps are the duration where they spend most of the time. Okay, there is a really interesting question that is there a pattern of pores that locks motile cell in a circular path or a back and forth motion? The short answer is we have not tested that yet. but it is possible to guide cellular motion because we have found that in a random network of pores, there exists loop-like motion or back and forth motion. But we are more interested in the randomness of the system, so that's what we studied. There was one question, can you explain the entropic trap? So we were inspired by this paper by Muthukumar, where he studies a trapped polymer chain in random porous media. Now, this polymer chain is governed by, thermal fluctuation. There are many orientation this polymer chain can take. However, there are only few orientation where this polymer chain can escape these traps. And there are many configuration where it will stay inside the pore. So if you take a log of those configuration, you get an entropy of the system, and you can gauge the time, how long that will spend in the trap. Now, we are making this up, and we say, like, we have an analogous system which where bacteria is inside this pore instead of this polymer. Okay, why my slide is. Okay, cool. But now, instead of thermal energy, the bacteria have their active energy. So their time spent should be a competition between this confinement and cellular activity instead of thermal motion. And since we also draw analogy from Bush's model of a thermally agitated particle moving through an exponentially distributed trap space, and since our pore sizes are also exponentially distributed, we think that the cells, we can use this model to explain the cellular motion through exponentially distributed trap confinements, where X is the competition between the confinement and cellular activity. So if we calculate this, we should find the trap time distribution should be power law distributed. And when we measured the trap time distribution in our system, we found that indeed they are power law distributed, and power is given by between -2 and -3. And I think I have covered the questions I wanted to cover. There are question and answer. So, Josh, how are you doing the question and answer? Are you going to answer it now, or we hold on, finish everything, and then go one by one?
Tapa, I've been answering them, like the ones that I can answer in the chat. You can type an answer. So I think like now that you're done, you can start.
I can go ahead and do that. You can start. Okay, yeah. That sounds great. Okay, cool.
Cool. So then I guess I'll move forward and start answering some questions as well. And annotate. Cool. All right. Hi, everyone. As I said earlier, my name is Jenna Ott. I am a G2 or a second year grad student in the Data Lab. My specialty includes bacteria forming what are called biofilms, which I'll touch on a little bit later on, as well as transport in lungs and more broadly, polymers in the body. And so we got some really good questions from you guys. There were a couple questions concerning the Howard Berg paper, so I'm going to go ahead and answer those questions for you guys. One of the questions is, does breaking off the filaments mean that they are dissolved and removed? This is relating back to the E. coli and removing their flagella. So in this case, not necessarily. So flagella can be broken off and removed chemically. They can go through different protocols to remove them. However, in this case, they're being removed through shear. You can see here a quote from the Howard Berg paper. Because shearing a dense cell suspension causes the filaments to tangle and fracture, we can break off most of a cell's flagellar filaments and then cement the cell body to a glass slide. So in this case, they're basically shearing off the flagella. So if we have an E. coli and we put it between either glass slides, or you can put them in a centrifuge and you shear, you can remove some of the flagella. And so in this case, they talk about using the shorter ends of the flagella to cement them to the glass slide. So that's kind of what's going on here. And there are a lot of different protocols that you can look up on how to do this, and it depends on what kind of strain, bacteria strain you're working with. And so basically you can remove different flagella and pili using different methods. And there are also strains of E. coli where they have been mutated such that they don't have flagella. So depending on what you're doing, that will determine what kind of E. coli you're using. And so the next question is, how do we cement the body to the glass slide? So this was a great question. In this case, what Howard Berg was talking about was some previous work done where they tethered the flagella from the E. coli to the glass slide. So I have put a citation here, paper 3. This is where they talk about this more in depth, but basically they shear off parts of the flagella so that way they're really short, and then they're able to tether them to the glass slide. And this is similar to the next question. Basically, why they want to tether it to the glass slide is so that way the cell is sticking to the slide, and then while they perform the rotation, they can look at the rotor because the flagella is tethered to the glass slide. And so the last question is, how do we attach a latex bead to the flagellar stub? And basically this comes down to a mutation in the flagella. They are made of different, here we go. They have repeating binding epitopes with an ionic charge, so they are very, what we would call sticky. And so they can stick to latex beads, which then they can use to look at the rotation, or they can even stick to glass slides. So that's just, you can mutate E. coli so that way their flagella aren't sticky, so that is something that people do. And so the next question, moving away from Howard Berg's paper, is how can the bacteria be recovered from the hydrogel matrix to be interfaced with other bioassays? This is a super great question, and this is actually one of the best properties of our experimental system. So if you remember from the from our soft matter paper, you can see the system here. And basically the hydrogels themselves, so each of these individual circles are not stuck to each other. This means they behave more like a ball pit rather than a solid matrix. And because it's a ball pit, we can actually consider this medium self-healing. And what do I mean by self-healing? This basically means that I can insert a nozzle into this medium. And by nozzle, I mean a needle or something like this, where I can retrieve something from this media. Maybe it's a cell, maybe it's the media around the cell to like look at the concentration of molecules in the media. But basically, if I can put the nozzle in the media, once I remove it, the ball pit will collapse again, so there will not be any destruction of the media. If you imagine like when kids jump in a ball pit and then they come back out, all the balls collapse into that open space. So we would definitely be able to recover the bacteria from this media, and that means that we can do other bioassays on the cells after we have put them in the porous media. Oh no, I forgot to put an animation. Sorry, guys. The next question is, what are the uses of bacteria hopping in our everyday lives and in industry? And this is my favorite question. I was so excited when I saw this question because the answer is literally everything. For me personally, I'm fascinated by the body and biology and health. And so the first thing that I think of when I think of hopping and trapping in our everyday lives is actually our lungs. And why do I think of our lungs? It's because our lungs are made up of, our lung mucus specifically is made up of biological polymers, and it actually is considered a polymer solution. And I think we are all astutely aware of the effect of different bacteria and viruses in our lungs right now with COVID. And so we actually have bacteria in our lungs all the time. Even if you're healthy, you have bacteria in your lungs. And so ideally they will be hopping and moving through your lungs, and that is super relevant. And our mesh size in our lung mucus actually is between 100 and 200 nanometers. So we can kind of use our model of hopping and trapping to imagine bacteria swimming through a polymer solution. And so, as I said, we all have planktonic or like normal bacteria in our lung mucus. And it's when these bacteria stop moving and start interacting with each other and change from a planktonic state that you start to develop infection and become ill. And so this is a big study that people do, and that's looking at biofilms, which I kind of mentioned in the beginning of my talk. And I will talk about this a little bit later. And the lungs aren't the only part of the body where hopping and trapping is important. Additionally, we have the gut, where polymers are in the gut, both as normal biopolymers as well as dietary fibers. So you have, you know, healthy gut biome, like the good bacteria that you want in your gut that are swimming around, hopping and trapping in these polymers. And then, of course, you know, we've all had some sort of stomach flu or some sort of digestive issue where the bacteria are not working as they're supposed to, and something is wrong, which can lead to infection, sickness, or disease. So this is, of course, hopping and trapping is important here. Now, there are other parts of the body. Our entire body is basically a porous media. There is not very much of us that it can be considered liquid media. So these are just some examples, but this can be applied to most of our body. And though I'm less skilled when it comes to these things, it's not just our body where bacteria hopping and trapping is important. There's also bacteria in our soil. So this has to do with, you know, bacteria in soil moving around plants, and then scaling this up to an industrial scale, we have bioremediation, which if you've heard of that, that's using bacteria to break down contaminants in dirty water that, you know, might be affecting the population. So there is plenty of uses of bacteria hopping and trapping, both within ourself, in our body, as well as in industry and our environment. And of course, there are plenty of other examples, but I just kind of wanted to touch on these. And so I'm going to give you guys some quick background before I answer the final couple of questions. I don't know if you all have heard of quorum sensing, but basically bacteria can secrete molecules called autoinducers. And these autoinducers allow the bacteria to kind of communicate with each other and with other species of bacteria. And so kind of how we can think of this is that as autoinducer accumulates or is not accumulating, this can trigger behavioral changes in the bacteria population. So that's just some simple background that you need for the next couple of questions. But we had some great questions here. One is, what kind of process would be used to influence a cell or bacteria to do something specific in an organism? For example, how do you tell an already existing microorganism or cell to do something different from what it's originally programmed to do? So this is kind of touching back on a normal planktonic cell in a healthy individual and what triggers it to become virulent such that you get sick. Because most of the time, the bacteria that cause illnesses in us every day, we normally have them in our body. So what triggers them to suddenly infect parts of our body? And so basically what's going on here is, if you see, I have this little bacteria guy on the screen. And if the bacteria are releasing autoinducer molecules, They keep releasing them. Maybe there are other cells in the vicinity, so all of the cells are releasing autoinducers, and so it's accumulating. And at some threshold concentration, this will trigger a behavioral change in the cell. Here I've shown the cell stops its motility and starts producing EPS. This is a sign that cells are starting to form a biofilm, but obviously this can trigger a range of behaviors such as turning on virulence, turning off virulence, changing growth, or changing consumption. There are so many different things that autoinducers can trigger in a cell. But basically, that would be one way to change the behavior in an organism, is changing how autoinducer is accumulating in the organism. And as I said, it can change a variety of different behaviors in the cell. All right, and the next question is, have we been able to use the hopping and trapping model to predict how infections spread through a body yet? And basically, whoever asked this question, great job. It's almost like you time traveled and saw what I was going to submit for my PhD thesis. One thing that we are working on right now that I can't tell you too much about, but is very exciting, is looking at a way to understand how the autoinducer accumulates in porous media, and looking at how this changes using simulations. And so that's kind of what we're working on right now. So if this is something that interests you, just remember Data Lab for the next couple of years and start looking for those papers. And I think this is my final question, which is, when two motile cells meet, how do they behave? Do they tend to move off in opposite directions, attempt to eat one another, or virtually ignore each other? And as I have talked about throughout these past couple slides, is bacteria can interact via autoinducer molecules. So this can cause behavioral changes among the population. They can also interact with different bacteria species. So if you have two motile cells from different species, they can actually interact also with autoinducer molecules and figure out how to kind of live together in harmony, as long as they aren't competing for the exact same nutrient, of course. And aside from autoinducers, bacteria can also move in collective motion in porous media. So this is something that Sujit is going to talk about a little bit in his talk. So that could be the case where they are moving kind of in the same direction, such as through chemotaxis, or something like this, so they can have similar behaviors. And finally, they will not, the bacteria, when they are hopping through porous media, they will not necessarily repel each other, but there is a huge influence in cellular crowding in terms of slowing down movement by bacteria. So if you have a not dense population of cells in the same porous media as a dense population, that dense population will diffuse and hop and trap much slower because they have the effects of crowding and bumping into each other. And that's it for me. Thank you guys for listening. I hope I answered some of your questions.
And I can aim to leave a little time at the end to just scroll through the questions we didn't get to, and we can answer those verbally. So I won't try and take too long with my slides. I'm having Jenna help me out because I'm out in the countryside with questionable Wi-Fi. So I'll just have Jenna scrolling through for me. So, okay. This particular question, motility of bacteria has many robust actions as ones that appear easy to model. Is there any way to predict the movement accurately, or is it more of a follow and map approach? This is a great question because it gets at the core of how is science done in general. And there's no one perfect answer because both occur. You look at what you see visually and then under a microscope, and then you try and understand it with a model. But sometimes people make predictions and then see if they work out. And so one notable example where someone made a prediction and saw if it worked out that will be talked about in Sujit's talk later is this paper, Crows, that was written by Crows in 2011. where they made a prediction that in a porous material where you have run and tumble bacteria, the collisions with the walls would cause more tumbling. And so the reorientation that tumbles describe could be mapped directly to the reorientation that collisions force the cells to undergo. And so they did out the math, assuming that every collision was kind of like a tumble, and they made predictions, but they didn't have the ability to see individual cells in the porous material. And so their results were based on theory that was well motivated, but they didn't have one crucial piece of information, which is what we found in Tapa's paper, where if you hit a wall, you stop. You don't immediately reorient. And understanding that that timescale was important was something that we found just by looking at it. And so this is an example where both sort of come together and have a role in moving the science forward. Can we go to the next slide? So do E. coli only detect aspartate? What other chemicals do they detect, and why those ones specifically? So it's interesting to note that the two receptors that you hear about a lot, TSR and TAR, they're named for the amino acid they respond most strongly to, but they're not exclusive to those that amino acid. So TAR is named for aspartate and TSR for serine, but they actually both sense a number of different amino acids. It's just those are the main two receptors, and they are not super specific is the point. But cells respond to oxygen depending on if they're aerobic or anaerobic, and if they're facultative, their oxygen response might change. And the cells can also respond to temperature, what's called thermotaxis. But the main question here is kind of why are they choosing to chemotax towards some things stronger than others? So I went to one of my favorite papers for this. Could you scroll down? Where a group under Viktor Storjic really explores this idea of, are cells truly attracted to the things that they can consume most easily and find the most tasty? And so I'll spare you the entire paragraph summary, which I found useful, but I can walk you through it in the next slide with the key result, which is they looked at the response to various amino acids and measured how sensitive cells were to the detection of these amino acids. So at a lower concentration for the one on the far left, each one of these graphs represents a different amino acid. And we're looking at, they looked at E. coli and B. subtilis. This one's for B. subtilis. And they looked at how much of that particular nutrient or attractant had to be in the, or here I guess I should call it an attractant because we're looking at the sensitivity, even though it's both. And they looked at how much activity the cell was producing in response to that amount of that attractant. And you can see that there's a whole range that spans many decades of concentration. So certain amino acids are very sensitive. The cell is very sensitive to, while others, it's less quickly going to respond. And then separately, in a well-mixed environment, they put they put the same amino acids in a vat with the cells and just watched which amino acids get consumed first. And then they tried to correlate these two to say, are they responding most strongly to the ones they consume first? Which is a claim that was in the Howard Berg paper, but only is really being followed up with here in a quantitative way. And so the next slide shows what that result looks like, where you try and correlate the midpoint of these two responses, so the 50% of relative kinase activity or 50% of initial concentration, the concentration where that happens and the time when that happens are these output measures of what is the consumption preference and what is the detection sensitivity. And for E. coli, they're correlated. There's a slope here going up. So if you're more likely to want to detect something at a low concentration, you're also more likely to eat it first. And then for B. subtilis, they found that it wasn't correlated, where this line was flat. So the next slide shows that. And this shows that maybe that hypothesis is not true in all cases. Maybe B. subtilis is looking for, quote, other environmental cues. So it's still an open question. So this is something that's going to be talked about in the talk, but it doesn't hurt to go over it again very clearly. Brownian motion is when something moves randomly. And so when something moves randomly, it can be described by diffusion, which is this equation, which just says that when things are moving randomly, they kind of want to spread out and fill in and get rid of any dense regions that are near not dense regions. And so we can think of this type of motion as composed of small straight line segments and reorientations, even for like pollen being buffeted around by collisions on the surface of water, which is the context where Brownian motion was first described. The collisions with other particles just sort of jiggle around in the pollen particles in small punctual little motions. And so there's a time scale associated with each part of this motion, the moving part and the reorienting part. And those time scales and how they combine goes into the diffusion coefficient D in the equation for diffusion. And so there's an assumption here that I just told you, which is that when you're moving in a straight line, you're moving in a straight line. It's sort of, you're not changing direction along those arrows. But this next question kind of gets at a deeper level of what does it mean to be moving in a straight line? It's how can we quantify, observe the rotational diffusive motion of bacteria? And while the cells are moving in a straight line, they're still undergoing thermal diffusion, which is sort of pushing them a little bit in different directions. So their straight lines might be a little curved, but a little imperfect because they're just having molecular collisions with whatever medium they're in. But that's not as strong a driving force forward as their run. So they're generally moving in a straight line. But after a while, if you're rotating by these small collisions, eventually you're going to change directions entirely if you wait long enough. So there's a way to characterize how short of a time scale you can look at where you can assume that it's practically straight line motion. And so the way to quantify this is to look at how much your angle has changed as you compare time step 1 to time step t, and then scan time step t along the entirety of the data you have. And so by looking at the square of this displacement in direction, or the change in direction against time, where I've made a mistake and made it log time, where I should either have it be log or not log either, on either. So as long as you average over enough paths, you can plot the change in position squared versus time, and if that has a slope of 1, then it's rotationally diffusive. And the intercept gives you a way to calculate that diffusion coefficient. So it's just like a translational diffusion coefficient, except it describes the direction that you're facing. So could you explain the difference between running and run and tumble movement and hopping in the case of movement in a porous medium? The difference is that the arrows are smaller. When you're colliding with solid walls, your runs that you want to take as 20 to 40 microns get stopped when you hit a wall. And so your run becomes only 7 microns or 2 microns. And so the difference between runs and hops, I would say, is just the size of how large these arrows are in this random walk. This is another topic that Sujit will go over, but it's difficult to wrap one's head around, so it doesn't hurt to go over it twice. What mechanism leads to the forward bias and the random walk in E. coli? So when you're doing run and tumble motion, you can modulate how frequently you tumble. But what that equates to is cells are able to elongate the runs when the arrow is pointing roughly in the direction of the gradient. So if we compare the left side and the right side, the bottom arrow is longer on the right because it's going to the right direction. Meanwhile, the arrow that's going straight down is roughly the same size because it's not going along the attractant gradient at all. And I included some smaller arrows going in the opposite direction, which has been observed to a lesser extent than the elongation of runs. The shortening of runs has been observed down a attracting gradient in this Saragosti paper that I will reference again in a minute. So that's the main way that cells generate biased motion, is they change how long they spend moving in one direction. I guess I forgot to just say quickly that the overall displacement from the start of this pattern to the end of this pattern is more to the right, on the right side. And that's sort of the point, is that by changing the size of these arrows, but not the direction of these arrows, I've managed to show that you can go more to the right. Thanks, Jenna. Okay, so there's another more subtle way that cells can bias their motion on average. And this is by changing the angle that they change, the degree to which they change their direction upon tumbling. So instead of taking this starting random walk, imagine that there's a gradient and you want to change direction less when you happen to move up the gradient in the right direction. So go ahead and scroll through pretty quickly, Jenna. I realize this is unfortunate that I'm having to have you scroll through this. It's one after another. So instead of changing directions to go further back left, that first tumble only changes directions a little less, and now you're making more progress forward than you were before. You can just scroll through to the end. Thanks. And so now you've got some biased motion to the right that you didn't have before, because you didn't reorient too much when you didn't want to. And so what this amounts to experimentally is that more of these arrows aren't longer in the right direction. More of the arrows are just in the right direction. So if you look at the probability distribution of the arrows, you're going to find more of them in that green section up the gradient, and fewer of them down in that red section down the gradient. And so we believe that the mechanism that causes this is related to this tumble bias that caused the other mechanism, but instead of changing the frequency of your tumbles, you're changing the strength, and you're doing that by changing the number of filaments, flagellar filaments, that are coming out of the bundle during a tumble. Because only one filament leaving will still reorient you a little bit. But if you have all of them go randomly, you're going to change directions more drastically. And so here they're showing the change in direction as a function of how many flagella were part of that tumble event. And the direction change was increased for more flagella being involved in reorienting. I believe that's all I've got there. And so maybe I can do my best to just answer some questions that didn't get addressed at this point.
Could I just jump in for a quick second? Hi, everyone. I'm Sujit. Daniel, Jenna, Tapa, thank you for just doing such an amazing job addressing those questions. So if I understand correctly, you have now addressed all the questions that were submitted. Is that correct? And now you want to jump to any other questions that arose in the Q&A, right?
Yep.
Can I just jump in for 1 second and ask if, and this is a complete surprise to all of you, but I was just thinking, like, so I'll be giving a lecture at noon on some of this stuff, highlighting the work that Daniel, Jenna, and Tapa have done. But I thought it might be nice for the students to get to know Daniel, Jenna, and Tapa a little bit. So do you guys mind just like giving a very brief bio of like who you are and how you got interested in the subject, just so people who know who you are, and then we can keep going with answering the questions. Is that okay? And maybe let's just go in reverse order of the people who just presented. So Daniel, why don't you go first? And I realize I'm putting you on the spot, but just tell people about who you are, where you came from, how you got to where you are now, and then Jenna, and then Tapa. And then you guys keep addressing the questions. What do you think?
Sounds good.
Cool, thank you.
So I'm from Pennsylvania, and I went to school at USC in Los Angeles. And while I was there, I worked in Professor Andrea Armani's lab, and sort of got into all the cool ways that scientists and biologists use imaging to probe different things. And imaging involves all these cool, very clever ideas from physics to utilize them in neat ways. And then, so that made me interested in pursuing a PhD and doing more research. And I thought I wanted to be a professor, but I'm sort of changing my views on that as I go. I'm sort of figuring that out a bit myself. And so I loved the physics of imaging, and I joined a lab where confocal microscopy is a big thing. So Sujit's lab, we love the confocal microscope. And I sort of, in this context, developed a real interest in modeling these continuum equations that describe chemotaxis. And the process of constructing a code that can model things and trying to make it fit real life with all the complexities of things that other models haven't incorporated has been really rewarding and fun. And it's inspired me to do more modeling outside of the context of bacteria stuff, but just in soft matter in general. And so I've been getting involved in more projects around the lab, just sort of exploring the ways that you can simplify a system and still get the key features of it. And that's a very engaging question for me. And there's all sorts of cool things with two-phase flow and porous media that are very fun to think about as a network model to try and extrapolate the interesting details of. I think that covers it for me.
Well, as I said earlier, my name is Jenna. I'm a second-year grad student in Sujit's lab. I'm originally from the Bay Area in California, and I went to school at UC Santa Barbara for my degree in chemical engineering. Within the lab, I'm kind of all over the place. I'm like 50-50 experimental and simulations at this point. With a lot of the simulations, I work a lot with Daniel on modeling bacteria in a continuum model. And for me, I focus on looking at biofilm formation. And in terms of experiments, I really like understanding aggregation of colloids or particles while under some type of shear. So this has a lot of applications, and it's a lot of complex physics. But right now I'm working on a joint project with some other people at Princeton to look at the aggregation of nanoparticles as therapeutics and looking at them under flow. So that's kind of where I'm at. I really like focusing on body. I think it's very interesting. And my long-term goal in life is to have all medication delivered through the lungs. So that way we don't have to get shots anymore. Specifically for an EpiPen, if we could deliver it through an inhaler, that would be great for all my food allergy people out there. And yeah, that's about it for me.
All right. Hi, I'm Tapa. My name is Tapomoy Bhattacharjee. I go by Tapa. I'm a postdoc at Sujit's lab. Before joining Princeton about 2 years ago as a postdoc, I did my PhD at University of Florida. Yes, go Gators. I did my work at Professor Thomas Angelini's lab, or known as Tommy Angelini. So when I joined Tommy's lab, I was a young grad student. Oh, I'm originally from India, so I grew up in Calcutta or Kolkata, and I did my undergrad back in India at Jabalpur University in chemical engineering. So I joined Tommy's lab in mechanical engineering to do my PhD. And I was fascinated by the idea that you can think of cells as like mechanical pumps. That's what his research was back then. So I started working with him, but we wanted to understand cellular mechanics in 3 dimension. So we got interested into how we create 3D structures of cells, of mammalian cells. And that led to a project which is 3D printing cellular communities, or bioprinting soft cellular communities, or in general 3D printing of soft material. That's what my thesis was developing a 3D printing process and studying cellular mechanics in 3D, or how cells move in 3D, developing a 3D culture media. So when I came to Princeton, I started working with Sujit. Sujit has an expertise on porous media. So he had a huge deal of work in porous media, which he did throughout his PhD and postdoc. So we completely clicked, and we started looking into how bacteria move through this soft three-dimensional porous media and how we can 3D print bacterial communities. That's what I focus now. I plan to be a professor and sometime soon, probably. And my goal is to combine my ideas in 3D printing cellular communities and studying biophysics problems to understand how cells interact in 3D dimension. I think that's it for me.
Awesome. Maybe you guys can keep going with the Q&A until we're done. I think we're wrapping up pretty soon, just a few minutes left.
You guys have such good questions. Oh my gosh.
So since we have like 1 minute left on the schedule, is there a way to save these questions and get back afterwards?
Yeah, maybe since Lana is recording this session, maybe we can get a copy of the questions that haven't been addressed and address them afterwards.
Yes, we will be running the report or all the questions. We will go through them, and they will be emailed to all the participants who attend the seminar.
Beautiful. So maybe just in the interest of time, maybe since it's basically 11, maybe we should wrap it up here, Lana, and then reconvene at noon for my lecture.
Sounds wonderful.
Daniel, Jenna, Tapa, thank you, and thank you to all the students who submitted such amazing questions. I'll see you everyone in an hour.
See you. Bye.